Thank you for attending our webinar on TruePrime, a unique primer-free MDA technology for genomic DNA amplification from single cells and cell-free DNA. Your speaker today is Dr. Angel Pitcher, our Director of Genomic Research, who has over 20 years' experience in this field. We will finish with a Q&A session, so if you have any questions, please type them in the comments section and we will answer as many as possible. If we don't get a chance to answer them, we will respond to them individually afterwards. So now let's welcome your speaker, Angel.
Thank you for the nice introduction. I would like to thank all of you for joining us today in this webinar about TruePrime. I will be covering the main advantages of this DNA amplification technology and how we have adapted TruePrime to the amplification of cell-free DNA derived from liquid biopsies. First of all, I would like to introduce TTH-Primpol, the key enzyme to develop the TruePrime DNA amplification technology. TTH-Primpol is a small member of the Primpol family of enzymes that contains the three conserved motifs A, B, and C that form the enzyme active site, but lacks the thin finger domain present in the other members of the family. In spite of this, TTH-Primpol shows the same properties than the other members of the family. As a polymerase, it can use both magnesium and manganese as metal cofactors. It shows DNA and RNA polymerase activities. It can be directed by DNA or RNA. It has certain strand displacement capacity. It shows high affinity for the NTPs. It is able to perform lesion bypass, especially in the case of 8-oxo-G, and it lacks terminal transferase and proofreading activities. As a primase, it can also use both magnesium and manganese as metal cofactors. It shows a convenient wide sequence specificity, but prefers the sequence CTC as the starting site. It can use ribose or deoxynucleotides at the 5' position, but only deoxynucleotides at the 3' position. Therefore, TTH-Primpol is a DNA primase, so we could envision a novel multiple displacement amplification method based on this enzyme. Previous MDA methods rely on the combination of Phi29 DNA polymerase and random synthetic primers. Depending on the input material, the method is called rolling circle amplification for circular molecules or whole genome amplification for linear ones. In TruePrime, we have combined Phi29 DNA polymerase with TTH-Primpol. In this setup, once the DNA is denatured, Primpol binds DNA at different recognition sites and generates DNA primers between 7 and 9 nucleotides long in a processive way. Then Phi29 DNA polymerase displaces Primpol and begins polymerization, performing strand displacement when the preceding strand is encountered. In those displaced strands, Primpol can bind again and generate new DNA primers that are again extended by Phi29 DNA polymerase, achieving the exponential and isothermal amplification. Expedeon has launched different kits based on the TruePrime technology. There's one kit available for the amplification of circular molecules, the rolling circle amplification kit, and two whole genome amplification kits to amplify genomic DNA that can be purified or directly from a single or a few cells. We have also two different solutions for the amplification of cell-free DNA obtained from liquid biopsies. The apoptotic cell-free DNA amplification kit is designed for the amplification of cell-free DNA derived from the apoptosis cell death mechanism, while the necrotic exosomal kit amplifies the larger fragments derived from necrosis or contained in extracellular vesicles, like the exosomes. TruePrime is able to efficiently amplify from lower DNA inputs than MDA methods based on random primers, showing an efficient amplification from just 1 femtogram of human genomic DNA. On the other hand, random primers require 100-fold more DNA to achieve the same amplification yield. The sequencing data obtained from the amplified material indicate that all the amplification products are target-derived when using TruePrime, even down to only 1 femtogram of input DNA in the amplification reaction. However, in the same conditions, the use of random primers results in a 20% of unknown sequences due to the primer artifacts that are generated during the amplification. TruePrime is able to amplify the whole genome from a single cell. In this example, 3 different cell lines were tested, showing an excellent reproducibility and linearity at the 3 different reaction times. To check the quality of the amplified DNA obtained from a single hex cell, we ran massive parallel sequencing with different number of Illumina reads. First, we compared TruePrime with methods based on random primers, having the non-amplified DNA from the same cell line as a control. The commercial method is Repligy from QIAGEN, and the generic method is just TruePrime without TTH primpole, but adding random synthetic primers instead. With 12 million read pairs, 53% of the human genome is covered with the non-amplified control. This represents a deviation of around 10% from the ideal coverage according to a Poisson distribution of reads. These are the coverage values obtained in each amplification case. As you can see, TruePrime shows the best coverage value and the lowest deviation from the ideal coverage possible, very similar to the values observed in the non-amplified control. However, on the other hand, both methods based on random primers showed much lower coverage and higher deviation. If we analyze the coverage chromosome by chromosome, you can observe how the coverage obtained with the commercial random prime reaction, here depicted in green, is far away from the values obtained with the non-amplified DNA in all the chromosomes, shown in orange. However, in the case of TruePrime, here in blue, the values are very similar to the non-amplified control in most of the cases, apart from chromosome 19 and chromosome 22, probably due to a high GC content. If we look at the coverage in chromosome 3, for example, the non-amplified material results in a very even coverage, although certain regions are overrepresented and others absent, due to the library prep or the sequencing, or both. TruePrime shows a profile very similar to the one obtained with the non-amplified DNA, highlighting the absence of any bias during the amplification process. On the other hand, the amplification methods based on random primers showed a much more spiky profile. The same happens in chromosome 4. Non-amplified DNA as a reference, TruePrime very similar to the non-amplified sample, and random primers showing a clear bias during the amplification. If we take a look in detail to a certain region, we can observe how similar is the coverage between TruePrime and the control DNA. Another example of this similarity in a pericentromeric region in which an overrepresentation event is observed and then no sequences are found in the centromere, but this is not due to the amplification step. The same performance is observed along the whole genome, as you can see in this circus plot. We also checked the reproducibility between TruePrime independent reactions using 5 million read pairs. Here you can observe the coverage in chromosome 3 and the profiles from 4 TruePrime independent reactions from single hex cells. The coverage is very even and very similar among them and in comparison to the referent DNA. And the same behavior is observed in chromosome 4. And again, this also happens along the whole genome, as you can see in this plot. We finally performed a deep sequencing experiment to detect single nucleotide and copy number variants. We compared the non-amplified DNA with TruePrime and Repligy. With a sequencing depth around 20x, we managed to cover 91% of the human genome in the TruePrime sample and the non-amplified DNA, very close to the limit of the Illumina technology. However, Repligy showed a much lower value. This slide shows the number of single nucleotide variants detected in each case, having TruePrime a value very close to the one obtained in the non-amplified control. This slide shows the correlation between different samples. The TruePrime sample shows 2.4 million SNVs in common with the control, while the commercial random primed MDA only 1.4 million. The comparison between the amplified and non-amplified samples allowed us to calculate the allelic dropout rates using different bioinformatic tools. As you can see, the allelic dropout rates are much lower in the case of TruePrime, independently of the tool used. Finally, TruePrime also shows very good performance to detect copy number variants, as you can see in this example from chromosome 6, while Repligy did not allow to detect any copy number variant. As a summary, these are the main advantages of the TruePrime technology. It is a very easy and reliable method. It doesn't generate primer artifacts. It shows an excellent sensitivity, down to femtograms of DNA. It shows very good reproducibility and very low bias in genome coverage. And it is suited for different next-generation sequencing platforms like Illumina and Ion Torrent. If you want to learn more about TruePrime, you can check the paper we published in Nature Communications at the end of 2016. It is open access. Let's move now to liquid biopsies. This term refers to the analysis of a sample of any bodily fluid, that could be blood, could be urine, saliva, etc., to detect molecular biomarkers that enable the discovery of a wide range of information about a disease or a tumor without the need for costly and invasive procedures. These molecular biomarkers can be circulating cell-free DNA, circulating tumor cells, circulating RNAs, but also protein markers and extracellular vesicles like the exosomes, for example, that contain DNA and RNA. In this slide, I'm going to summarize the workflow for the analysis of cell-free DNA samples obtained from liquid biopsies. First, from a liquid biopsy sample like a tube of blood, plasma can be isolated by centrifugation. DNA can be purified from plasma, including cell-free DNA derived from healthy and tumor cells, but also DNA derived from exosomes released by healthy and tumor cells, resulting in a mixture of different DNA molecules from different origins. However, the amount of DNA extracted from these samples is frequently very limited and not enough to perform different analyses. Therefore, we propose to use the TruePrime DNA amplification solutions for liquid biopsy samples to generate enough DNA material to run different analytical methods and techniques, like the next generation sequencing or qPCR and digital PCR, in order to enable the promises of liquid biopsies that are early diagnosis, improved prognosis, personalized therapeutics, and more sensitive disease monitoring. There is an intense discussion in the field regarding the different mechanisms that originate the cell-free DNA found in blood or any other bodily fluids. In healthy cells, the main mechanism is apoptosis, in which the DNA is cut between nucleosomes, generating short DNA fragments between 160 and 170 base pairs. However, in cancer cells, other mechanisms also occur, like necrosis, producing different DNA molecules in terms of size. Finally, living, healthy, and cancer cells release big DNA molecules contained in exosomes, for example. Expedeon has developed different solutions depending on the size of the target DNA molecule. In the case of the short cell-free DNA molecules derived from the apoptosis mechanism, we have available the TruePrime apoptotic cell-free DNA amplification kit. If you are interested in the larger DNA fragments derived from the different mechanisms or contained in extracellular vesicles, Expedeon has a different kit for this amplification, the TruePrime necrotic cell-free exosomal DNA amplification kit. First, I'm going to explain the apoptotic cell-free DNA amplification technology. The purified apoptotic cell-free DNA fraction may be degraded or damaged, having protruding 5' or 3' ends, and lacking 5' phosphate or 3' OH groups. For this reason, the first step of the protocol consists of an end repair reaction to fix those lesions. Moreover, 3' ends are tailed with a protruding A. The maximum sample volume in this step is 50 microliters, which is the typical elution volume from most of the cell-free DNA purification kits, so you can use the whole elution volume for the amplification reaction. The next step is the adaptor ligation to both ends of each cell-free DNA molecule. The adaptor is a 36-mer oligonucleotide that self-hybridizes, forming a hairpin structure having a 3' protruding T. This allows the efficient ligation to both ends of each cell-free DNA molecule. There is no purification step between end repair and ligation to minimize the DNA loss during the process. After ligation, the DNA is a bit-based purification is required to remove the remaining adaptors and the enzymes from previous steps. The DNA is eluted in 15 microliters that are used directly as input in the amplification reaction. The adapter hairpin structure contains the preferred binding site of TTH Primple, the sequence CTC, so the DNA primer synthesis begins from both ends of each DNA molecule, independently of the sequence of the cell-free DNA fragment. The DNA primers synthesized by TTH Primple are recognized and extended by Fi29 DNA polymerase, which results in multiple concatenated copies of the original molecule. Those copies contain also the adapter sequence and structure, so new priming events occur, achieving the exponential amplification of cell-free DNA. The TruePrime apoptotic cell-free DNA amplification kit shows an excellent sensitivity and efficiency, achieving DNA amplification yields in the range of micrograms, starting from picograms of cell-free DNA. It is remarkable the proportionality between the input amount and the amplification yield observed. In this slide, you can see the amplification yields obtained from 1 nanogram of cell-free DNA from 48 different cancer patient samples. The amplification yields are very similar in all the cases, independently of the tumor type and grade. We have run two different sequencing approaches and platforms to validate the use of this amplification technology. First, we ran low-pass whole genome sequencing using Illumina per N reads. And we also ran targeted sequencing using the Oncomine panel for colon cancer and ion torrent sequencing. We selected one colon cancer patient for low-pass whole genome sequencing. First, we analyzed the cell-free DNA size using the Bioanalyzer high sensitivity chip, and we observed a very nice mononucleosomal peak, as well as a smaller dinucleosomal peak. We amplified 4 nanograms of cell-free DNA in 2 independent reactions and sequenced the amplified material in parallel. As you can see in this plot, we observed a very even coverage along the genome that allowed us to detect several copy number variants. You can see in this slide the excellent reproducibility between the 2 independent amplification reactions, zooming in at different scales. As I mentioned before, for the targeted sequencing we selected the Oncomine colon cell-free DNA assay from Thermo. This panel enables the analysis of single nucleotide variants and short indels in 14 genes, and more than 240 hotspots are covered. 3 colon cancer patients were included in this assay, in which we compared the amplified and non-amplified cell-free DNA using different DNA inputs, as you can see in the last 2 rows of the table. As in the case of the Illumina sequencing, we first analyzed the cell-free DNA size using the Bioanalyzer high sensitivity chip, and we observed clear mononucleosomal peaks in the 3 cases. After sequencing, We ran variant calling for single nucleotide variants, multiple nucleotide variants, insertion and deletions at different frequencies, from 35% to 1%. In the 3 patients, we observed more variants in the amplified sample than in the non-amplified one, independently of the mutation allele frequency threshold. Moreover, the number of annotated variants is also higher in the amplified samples than in the non-amplified ones. In this slide, you can see in more detail the variants detected in the samples derived from the first patient. The number of clinically relevant variants is higher in the amplified sample, increasing the sensitivity of the analysis. For the 1% frequency, 30 clean bar mutations are detected in the amplified sample, while only 14 in the non-amplified one. From those 14, 10 are also detected in the amplified sample. For the 35% frequency, 3 clean bar mutations are detected in the non-amplified sample. Those 3 and 2 additional clean bar mutations are found in the amplified material. In the second patient, the number of clean bar mutations is also higher in the amplified sample. For the 1% frequency, 31 clean bar mutations are detected in the amplified sample, while only 13 in the non-amplified one. From those 13, 10 are also found in the amplified sample. In the third patient, the number of clean bar mutations is again higher in the amplified sample. For the 1% frequency, 26 clean bar mutations are detected in the amplified sample, while only 16 in the non-amplified one. And from those 16, 13 are also covered in the amplified sample. Therefore, the amplification step before sequencing increases the sensitivity of the analysis, enabling the detection of more clinically relevant variants. Now I'm going to guide you through the data we have generated using the necrotic cell-free exosomal DNA amplification kit. I'm going to show the results we have obtained with 2 different tumor samples and 2 different sequencing approaches, targeted and whole exome sequencing. We first checked the presence of mononucleosomal cell-free DNA using the tape station. As you can see, there's no mononucleosomal DNA in the first case and only a very small fraction in the second one. However, the 1.5 kb marker in both cases seems to be larger than expected. So we ran the same analysis using the Bioanalyzer high sensitivity chip to increase the size of the fragments that could be detected. We then observed long DNA fragments next to the 10 kb marker in both cases. Our hypothesis is that these long DNA fragments are not necessarily contaminating genomic DNA from blood nucleated cells occurring during plasma isolation. They can be derived from necrotic cell death or extracellular vesicles containing DNA, and could be more related to cancer than the apoptotic fraction, which is mainly derived from healthy cells. These long DNA molecules are the ones specifically amplified with this kit. These are the yields of the amplification using 100 picograms of cell-free DNA as input in the reaction. The targeted sequencing approach allowed us to detect single nucleotide variants in 50 genes related to cancer. As you can see, the amplified material shows more SNVs than the non-amplified cell-free DNA, in the range of the ones detected using DNA isolated from the tumor tissue. The correlation between the results obtained in each sample showed that the amplified cell-free DNA shares more SNVs with the tumor biopsy than the non-amplified cell-free DNA, highlighting the efficiency of the amplification process to amplify circulating tumor DNA specifically. This graph shows the number of common variants between the amplified cell-free DNA and the tumor biopsy in each gene of the panel. In the second case, we compared the TruePrime amplified cell-free DNA with the DNA derived from tumor tissue and healthy tissue, as well as the amplified material obtained directly from plasma without a cell-free DNA purification step. Unfortunately, as it is pretty common in these cases, we didn't obtain enough cell-free DNA from plasma to sequence the non-amplified material. The coverage is very good in all the cases, even in the sample amplified directly from plasma without DNA purification. There are several copy number alterations that can be detected in the amplified samples and the tumor tissue, but not in the healthy one, supporting the relation between the cell-free DNA fraction and the tumor. We checked the variants that could be detected in the same 50 genes related to cancer as we did in the targeted approach. As you can see, the correlation between the amplified material and the tumor biopsy is excellent in terms of single nucleotide variants, having more than 500 in common. Finally, I just want to review again the different TruePrime solutions we have depending on the target DNA molecules you want to study. You can select the TruePrime apoptotic DNA amplification kit to amplify the short DNA fragments derived from apoptosis, or the necrotic cell-free exosomal DNA amplification kit if you want to study the larger DNA fraction from your cell-free DNA prep. I'd like to thank the Expedeon genomics team and Luis Blanco's group from the Molecular Biology Center that helped us to characterize TTH Primple. I also want to thank the 2 hospitals we are collaborating with to develop the liquid biopsy kits. Finally, I want to stress our deepest gratitude to the cancer patients that kindly provided us with the samples required to develop and validate the application of TruePrime to the liquid biopsy field. Thank you so much for your attention. You can contact me by email if you have any question, and let's now answer a few questions received during the webinar. The first question is about the compatibility between the TruePrime amplified DNA with different NGS techniques. Yes, the amplified DNA is compatible with different NGS platforms. I have shown data using Illumina and Ion Torrent, but we have also used internally for other purposes PacBio and Oxford Nanopore sequencers. Obviously, you have to follow the specific library prep kits and protocols developed for each platform. Another question is, what is the length of the TruePrime amplification product obtained using the apoptotic DNA amplification kit? The size distribution of the amplified material will be between 2 and more than 50 kb, with peak between 15 and 20 kb, more or less. Another question is, what is the maximum amount of cell-free DNA that can be used for the TruePrime apoptotic cell-free DNA amplification kit? Well, you can use up to 150 nanograms of purified cell-free DNA as a starting material. That is quite a lot DNA. Another question is, do I need to clean up the TruePrime amplified DNA before downstream analysis? Yes, we strongly recommend to purify the DNA for further analysis, because some of the TruePrime components could be inhibitory for the downstream processes. You can use PCR purification kits, like the ones sold by QIAGEN, for example, or pit-based purification protocols. Both should work well. Finally, what is the fidelity of the TruePrime amplification techniques? Well, the fidelity of TruePrime relies on the use of Fi29 DNA polymerase, which is probably the polymerase with the highest fidelity due to the proofreading activity. And the extremely low error rate should be around 10 to minus 7, 10 to minus 8. So pretty good. Well, these are more or less the questions received. If we receive more questions, we will be answering those by email as soon as possible. So thank you again for your attention and for joining us today for this webinar about TruePrime and the solutions we have developed for the liquid biopsy field. Thank you.